Hola, bienvenidos. Me llamo Nick Keiser. Thank you for coming to my TikTok. I'm really excited to be presenting this research on behalf of Elise Richardson, who is now a PhD student at North Carolina State University, but she was a master's student in my lab at UF from 2019 to 2021. So I'm going to be presenting this research on her behalf, but I wanted to start by acknowledging the research team that worked on this collaboratively. So Sam Shablin is another graduate student in my lab who helped with statistical analyses. Caitlin Taylor works in the University of Florida Department of Entomology and helped collaborated with Elise on DNA extractions and sequencing. Brittany Jabot was an undergraduate at the time, but is now has a master's in public health. And she helped with basically everything. And Dr. Estelle Martin from UF Entomology helped with bioinformatics and sequencing as well, and experimental design. Okay, so I named the title of my talk Behavioral Parasitology purposefully, and that's because I think it is a phrase that should be in the lexicon of behavioral ecologists more than it is now, something that we should be familiar with. I think many of us, when we hear a phrase like behavioral parasitology, we probably think of the things that we learned in Janice Moore's book. You know, how do parasites alter host behavior, and how does the behavior of hosts influence, their likelihood of becoming infected with parasites, things like that. But that's actually not what I'm referring to here. I'm referring to the phrase that was coined by Kenneth Saladin in 1979, referring specifically to the behavior of parasitic animals. So as behaviorists, I think we're all familiar with, brood parasites and social parasites, which are very interesting and cool in their own right. But that's not what I'm talking about here. Here, we're talking about the types of parasites that eat you slowly. The type of parasites in a trophic ecology sense, they move energy through food webs. And this is a pretty overlooked aspect of animal behavior. These are animals and they behave, but I don't think many of us are familiar with this type of work. So in 1994, there was a special issue in the journal Parasitology titled Parasites and Behavior, which is exactly the type of place you would expect to find papers on parasite behavior. But instead, half of the papers in that special issue focused on the behavior of the host. In 2004, there was a review paper on trematode behavior, and the authors of this paper are basically arguing, in the life cycle of this huge diversity of parasites called trematodes, we know that there's these free living or free swimming stages, and they're behaving in those stages. They're finding new hosts. So it'd be really important for us to understand what is going on in those stages. And what I think is really interesting is they even, have to use these terms that we learn in intro animal behavior like clinotaxis and clinokinesis to really drive home the take-home message that yes, parasites have behavior. And I think this is really important to remember that, the majority of animal species that we've described are parasites. It's not like a small, rare thing. It's kind of the norm when it comes to lifestyles across the planet. And many of those parasites need to find their hosts, right? They're not just born on a host and then they start feeding on blood or something like that. Many of them do that, but many have to find new hosts after they hatch or after they're born. So I think it's important that we study how that process occurs. I think behavioral parasitology is particularly important for vector-borne diseases because transmission is dependent on vector behavior. So we're talking here, mosquitoes aren't exactly parasites in the truest definition, but when you think about mosquito-borne diseases or tick-borne diseases, ticks are actually parasites. They're behaving in certain ways to find new hosts. And ticks are number one when it comes to the diversity of pathogens that they transmit. They find their host via a behavior called questing. So they leave the leaf litter to crawl up onto a piece of vegetation, and they'll stick their arms out like this and wait for a host to walk by, and they'll latch on, and then that's where they get their first blood meal. Questing behavior is, it's pretty straightforward, but there are some complexities involved with it. And some really interesting work has found that questing behavior can be influenced by pathogen infection. So depending on the pathogens that a tick is carrying, their questing behavior can change. So I think it's really important that we should be studying that. Okay, so Lauren Cater and collaborators have laid this really awesome theoretical foundation that suggests that trait variation in disease vectors is really important for epidemiology. So here they're talking about biting probability, but it could be questing behavior in ticks as well. So they suggest that variation among individuals, variation over time, variation across different environmental gradients are all important in influencing disease dynamics, but we really have to understand them in sort of a joint process, in sort of a combined perspective to really understand how vector behavior can influence infectious diseases. So here we wanted to ask, how does pathogen infection influence tick host-seeking behavior across these different types of scales? So specifically, we were asking, do ticks exhibit individual differences in questing behavior? Does questing behavior differ between different habitat types that are nearby? Does questing behavior differ between populations? And how does pathogen infection affect questing behavior across some of those different scales? So we're explicitly trying to ask questions at the individual level, between different habitats, and between different populations. The tick that we were using for this research is the Lone Star tick, Amblyomma americanum. This is described as an aggressive biting tick, which means that humans often find it on themselves. It's a major vector for several diseases, disease-causing pathogens, including Rocky Mountain spotted fever, which is a rickettsial disease. Here we were focusing on Rickettsia amblyommatis, which is related to Rocky Mountain spotted fever, but we don't really know a whole lot about it. It's a newly emerging pathogen, and we don't know what its effects are on ticks. And what I think is really important in this context is that the Lone Star tick range is expanding throughout North America. So 50 years from now, there will be places that did not used to have ticks and now they do, and there will be places that used to have ticks and now they do not. So I think that's why we need to be studying tick behavior across different types of habitats. Okay, so the way that you collect ticks from the field is you use this device called a tick drag, which is basically just a canvas sheet with some rope attached to it, and you drag it through the forest floor or over vegetation, and it'll pick up any questing ticks that are on the vegetation that grab onto it, thinking it's a host. Then you grab the ticks, take them back to the lab, and study their behavior. So Elise developed this assay where you place a tick in a plastic container with a wooden dowel rod sticking out of the middle, and she would wait until they crawled up the dowel rod, and she would measure whether or not they quested, so yeah, a yes or no variable, the height at which they quested, and the duration of that quest in a 10-minute assay. To ask the first question, how does host-seeking behavior differ between individuals? She measured the same individual ticks repeatedly for 3 days in a row. So she did this with 280 ticks, measuring them 3 days consecutively. We would have wanted to go longer than that, but it's just hard to keep ticks alive and doing well in this type of setup. So, but what we found was that questing behavior is repeatable across those 3 days. So this graph shows the repeatability estimate and the 95% confidence intervals. So if the confidence intervals don't overlap 0, which is the dotted line, we would say that that behavior is repeatable. And by repeatable, we mean that there is more variation among individuals than there is within an individual over time. So if something like questing height or questing likelihood is significantly repeatable, this would suggest that ticks are consistent across time, but they're also consistently different from each other in this behavior. So we found that for 2 out of these 3 variables, but what we also found was that infection was a significant predictor of the repeatability of questing likelihood. So infected ticks were more repeatable in their behavior than uninfected ticks. I actually have no idea how to interpret this, so I'd really be interested to talk to anyone that think this is interesting and would like to chat about it. Okay, the second question, does host-seeking behavior differ across different habitats? So we collected ticks in 2 different habitats in the Ordway-Swisher Biological Station, which is a field station operated by UF. And so xeric hammocks have sort of a more open understory, and successional hardwoods have a more complex understory with more vegetation and coarse woody debris and things like that. Elise brought them back to a common garden area, which, because this experiment was conducted in 2020, was her patio in her apartment. So she brought all the ticks back to the same place and measured their behavior there. What she found was that ticks from xeric hammocks quested for 2.5 times longer than those collected from successional hardwoods. So we weren't testing their behavior in those habitats. We brought them back to a controlled environment, and we found this difference in behavior. So what I think how I interpret this is that if you are growing up in a habitat like a xeric hammock, where there's not much understory complexity, anytime you find an opportunity to quest, you should take that opportunity and make the most of it, because that's a really valuable opportunity. Whereas if you're in a successional hardwood that has all these opportunities and places to quest, then it's not as important and you can sort of quest for less time. Okay, so the last question, does host-seeking behavior differ between populations? So Elise collected ticks from 3 different parks, public parks in Gainesville, which is the city where the University of Florida is located. And she also collected ticks from rural sites in the Ordway-Swisher Biological Station. So this is very, rural. There's very little foot traffic, and it's, natural habitats. So there are about 40 kilometers between these sites. And what she found was that ticks from the rural sites quested about 7 to 12 centimeters higher compared to those from the 3 city parks. This is another result that I actually don't know how to interpret. I don't know what this means, so would love to chat about it. Okay, lastly, does infection status alter tick host-seeking behavior? So first, we found that adults were more likely to be infected, which makes sense because this is a multi-host tick. So it is taking multiple blood meals through its life cycle, experiencing multiple hosts, and having more opportunities to become infected compared to nymphs who are younger. But we also found that infection was associated with a 1.5 times shorter questing duration, but only in nymphs. So infected nymphs in this yellow color quested for less time compared to uninfected nymphs, but we did not find this effect in adults. So I think I'll say this for the 3rd time in this talk, I actually don't know what this means. I think that we don't know if it's driven by, I know what we don't know, and that is that we don't know if it's driven by the pathogen, if it's somehow altering the behavior of the host being the tick in this case, or if it's some sort of adaptive response of the tick to its own pathogen infection. We don't really know. If we could collect ticks, measure their behavior, infect them, and then measure their behavior again, that would really be the only way to test that. Right now, we just have this correlative evidence. So we found no effect of pathogen infection affecting behavior differently across different habitats or populations. So there was no complex interaction terms between infection status and habitat type or population, which is something I'll get back to in a moment. Okay, so our take-home messages are, at the individual level, Lone Star ticks exhibit individual differences in questing behavior. Ticks quested for longer in habitats with a more open understory. And ticks from rural sites quested at greater heights compared to ticks from city parks. And we found that infection with this rickettsial pathogen altered the repeatability of questing behavior and was associated with a reduced juvenile questing duration. So I think this is really important because we know at multiple, regional and global scales, disease vectors are shifting their geographic ranges in multiple different, sometimes predictable, sometimes unpredictable ways. And as vectors as their range expands, they're potentially inhabiting or interacting with novel habitats. So I think it's really important for us to know how their behavior, especially their host-seeking behavior, might change in those habitats if we really want to inform, epidemiological models or public health measures or something like that. So far, we didn't find any evidence that suggests that, the pathogens that they're carrying with them might affect their behavior differently in the new habitat than it did in the old one. But, this was only one pathogen and one vector. So I think it's something that we should need to study in more systems. So with that, I want to say thank you to my current lab members, my newest lab member, Miiha. This is Piper Keiser. All of the resources and funding sources that we had to do this work. And if you want to access the data, the raw data, or any of the code that I presented in this talk, you can use these QR codes right here. And thank you all for listening. Muchas gracias. Obrigado.